researchers replace resistant gene with a susceptible counterpart, opening the door to new methods that could fight malaria and reduce pesticide use. Insecticides play a central role in efforts to counter global impacts of mosquito-spread malaria and other diseases, which cause an estimated 750,000 deaths each year. These insect-specific chemicals, which cost more than $100 million to develop and bring to market, also are critical to controlling insect-driven crop damage that poses a challenge to food security. But in recent decades, many insects have genetically adapted to become less sensitive to the potency of insecticides. In Africa, where long-lasting insecticide-treated bed nets and indoor spraying are major weapons in the fight against malaria, many species of mosquitoes across the continent have developed insecticide resistance that reduces the efficacy of these key interventions. In certain areas, climate change is expected to exacerbate these problems. University of California San Diego biologists have now developed a method that reverses insecticide resistance using CRISPR-Cas9 technology. As described in the journal Nature Communications, researchers used the genetic editing tool to replace an insecticide-resistant gene in fruit flies with the normal insecticide-susceptible form, an achievement that could significantly reduce the amount of insecticides used. The researchers used a modified type of gene drive, a technology that uses CRISPR-Cas9 to cut genomes at targeted sites to spread specific genes throughout a population.